Welcome, Life Sciences. Welcome to another exciting life science lesson. We are going to recap the cell. We're going to go through all of the important parts, just hopefully all those terminology comes flying back, all those diagrams coming back to you. It's really important at the end of each section that you do go back. I'm hoping that you maybe have got summarized notes, that you have got summaries, that you've done little notes on the section, because obviously it's a really important section that you need to be able to do. It's one of the foundations for the rest of the sections that we're going to carry on with. Okay guys, so where did we start off? We looked at this whole concept that if we're going to look at cells, all right, we need to be able to see cells. Let me get my pen going over here. So how do we see cells? So if we look at technology over time, technology, all right, is enabled us to see the smallest things, and that is the microscope work. As I said, I hope that some of you do have access to microscopes, right, at your classroom. If not, it's unfortunate, but the whole concept of a microscope is that I can see something that's really small, I can see it bigger. And this is what has enabled us to see cells, right? To be able to see what they look like, to be able to tell us what they do, is this whole concept of technology. So throughout time, what have we been able to see? Microscopes have got better and better and better. Remember, you had the light microscope maybe at your school, then it's really big, or universities and laboratories, they've got transmission, all right, electron microscopes, and all right, your electron transmitting microscope, okay, your LEM, your TEM, and your SEM. So we've got, with this whole thing, we now have a better idea of what cells look like. Now, when we look at this concept of a cell, there's just two terms that we need to look at. A cell can be either a prokaryotic cell or a eukaryotic cell. And what do I mean by that is quite simply, if we look at a timeline, right, of throughout time, prokaryotic cells were one of the earliest living cells. They are unicellular, all right, unicellular. They are only one cell, yet they are able to, all right, do the whole, as I said, Mrs. Greef thing. They are able to undergo all the living characteristics that we looked at. They are quite primitive, right, in their being. They are oldest today, right? They are bacteria. They have the whole kingdom called, all right, the kingdom Monera, and they are prokaryotes. And simply what they are is they have a cell membrane, they have cytoplasm, but they do not have a nucleus, and that was very important, and they do not have, all right, any membrane-bound organelles. So they had a cell membrane, and they had cytoplasm, and they usually had some kind of DNA, and it was not in a membrane. Then as time went by, all right, organisms became more complex, and with that complexity, needed a cell that could do, all right, far more complex things. And hence that cell is the eukaryotic cell. Now, when we look at the eukaryotic cell, we are going to divide that cell into a plant cell and an animal cell. And remember, guys, you must be able to tell the difference between a plant cell and an animal cell. Now, when we looked at this concept of a cell, we noticed that all living cells, especially eukaryotic cells, have three basic things in place. Number one, they're all surrounded by a cell membrane, okay? And that cell membrane kept everything in and kept everything out. So you have this enclosure. Then we looked at this concept of cytoplasm, and as I say to you, you need to think of a jar of jelly, making a bowl of jelly. That's what cytoplasm was about. And then inside the cytoplasm, you are going to find structures, little structures that have got certain functions. Now, all the cells that we're going to look at, all of them have a nucleus. And we're going to have a look just now what that nucleus is. Simply, it means a structure by itself. It's separate to the cytoplasm, right? It's in the cytoplasm, but it has a membrane of its own. And most importantly, we're going to have a look just now. It's got that chromatin network where we carry our hereditary characteristics. So we started off by looking at what the cytoplasm is. And the cytoplasm, remember I said to you, is that jelly, right? So we take, there we go, the jelly, we take water, we take the jelly, we put it together, and that's what we're going to get. So in here, the most important constituent is water, right? Water makes up approximately 60% of all the cells. Then dissolved in the water, we're going to find gases and all our mineral salts. Now, also in the cytoplasm, not necessarily, right, dissolved, we're going to find our proteins, our carbohydrates, and our fats. Now, guys, remember the whole thing about is we eat food. We eat food because we need it for energy and we need it to build our bodies. So when I ask you about this concept of build, what does building mean? Building simply means we need to build cells. So the food that we eat, all right, goes towards making these cells work. We need to build them properly. If we build them properly, they are going to work properly. Now, when we looked at this concept again of a cell membrane, cells have got these boundaries. What is a boundary? It means the outside. And the reason why I've drawn a plant and an animal cell, or I've given a picture of a plant and animal cell here, is because plant and animal cells will have different boundaries. Now, if we have a look here, an animal cell has one boundary, and that is going to be the cell membrane. If I look on this side over here, I'm going to see that the plant cell also has a cell membrane. So all living cells are going to have the cell membrane. But the plant cell has an extra boundary on the side, and that is going to be the cell wall. Plants need a little bit of extra support. Plants need, all right, to keep their shape, and so they're going to need a cell wall. So if we have a look here at the structure of the plant cell, right, what is a cell wall? We only find it in a plant cell, very important. It allows everything in, and what's the most important component is cellulose, right? And if you can remember in your biochem, cellulose was a polysaccharide, right? So it's made up of, it's a carbohydrate, and it's made up of a saccharide, the polysaccharide, like starch and everything. And here we have the cell wall, nice and strong, non-living, to be able to support the cell. Plants, the cell wall also needs to have spaces in which they can communicate, and they do so via these little holes or these passageways called the plasmodesmata. Now, when it comes to the cell membrane is quite a complicated, all right, structure. And we give the name a fluid mosaic model. And what do we mean by that is the fluid means that the cell membrane can move. When we go to our plant cell, the cell wall over here, we notice it's very rigid. And another word I would use there is permeable. What do I mean by that? Is the cell wall is going to let everything through. However, when it comes to our cell membrane, we can say that our cell membrane is living, it's alive, right? It's a fluid molecule. It needs to be that because very often animal cells need to be able to move. What do we mean by a mosaic? there are structures, there are carbohydrates, there are proteins that fit into this fluid mosaic molecule, and these little phospholipids, the bilayer, and this whole structure of the cell membrane, right, is to facilitate, we're going to look at that just now, right, transport. The most important thing that the cell membrane does, although we know that it encloses, is it is selectively permeable. Whereas the cell wall will let everything in, this whole structure of the cell membrane allows things to move in and to move out, so we can cross, so we can control what we need to look at and what we need to. So what comes in, what comes out, we can have control over that. Okay guys, I'm afraid that's all we have time for now. We're going to have a quick break and we'll be right back.